Okay, this video is about enzyme active sites and your health. Okay, so basically an enzyme is something that catalyzes a chemical reaction to make it occur more rapidly. So these are the substrates, what you begin with. So here's substrate number 1, here's substrate number 2, and then they're going to fit together. This will be the transition state of the chemical reaction, and then the substrates will separate from each other in a new form where this will be now product number 1, product number 2. The classic description of enzyme and substrate is that the enzyme's like a lock and the substrate's like a key. And then as study of enzymes became more sophisticated, it was realized that it's a little bit more like a hand going into a glove, whereby as the substrates enter the enzyme active site, they change the shape of the glove a little bit, and that's called an induced fit. Okay, now a little bit more detail. The reason why this is worth knowing is a general understanding of some enzymes concepts is important because you're going to hear about them all the time in biochemistry, and this relates to nutrition and health. So first of all, if you've got the substrates in the enzyme active site and they're not facing each other, that's not going to work very well. The whole purpose of the enzyme is to accelerate the rate at which the reaction occurs. So it needs good proximity. It needs to have the right shape, the enzyme active site, to keep them crowded close together, substrate number 1 and number 2, so they will undergo a reaction. They have to be orientated correctly, so it sticks into, for example, they bind with each other, substrate number 1 and substrate number 2. Other things so we just talked about a lock and a key, then we talked about a hand and a glove, but now there's a third concept. We talked about proximity, we talked about orientation of an active site functioning effectively. There's also the issue of charge. And when there's a negative charge, for example, in an enzyme active site, if the substrate had a positive charge, that's like a magnet to pull the substrate into the active site. So these enzymes can react incredibly quickly. And this is how they do it. They're a perfect fit. They've got a magnet-like charge in the center of their active site that pulls the substrate in and then immediately puts them together. Reaction occurs and it lets them go. So remember that it's like a magnet. That's another good trick to remember about enzymes. And this stuff here is going to relate to receptors for all kinds of biochemistry stuff. All right, so what does an enzyme do in terms of how can you diagram the reaction? You start out with a substrate and then you end up with a product over here. Normally, it takes a lot of energy to get that transition state to occur, and it rarely occurs typically in regular life without the presence of the enzyme. Versus with the enzyme present, the reaction, the energy state of the energy level of the transition state is markedly lowered, which greatly facilitates the reaction and the production of the product. So here's the energy level, free energy level. Here's the reaction progress. Now we're getting to the good part here. Okay, now I'm just going to give you one theoretical example. Here is an amino acid called glycine. It's the smallest amino acid. In the center is the carbon called the alpha carbon. And then the alpha carbon for glycine has a side group of only an R. The R group is only a hydrogen. It's the smallest amino acid. There's always going to be an amino on one side of the alpha carbon. That's the amino of amino acid. And then there's a carboxylic acid. Carboxyl group is called here. That's the amino acid, the acid of the amino acid. Okay, so now forgetting about that one side of the page, let's look at glyphosate. Glyphosate is a herbicide, and it's one of the most commonly used herbicides. So basically, you got an alpha carbon here, and just coming off the alpha carbon is a single hydrogen. So it's the same as glycine in terms of this part of the molecule and this part. All of this is exactly the same as glycine. But attached to it is a methyl group with a phosphate group attached to that. So its full name would be like N-phosphomethylglycine. You could call this a methylphosphonal group. Okay, but this is a key point, that it is just like the amino acid except with this carbon phosphate attached to it. So this similarity on one side means, according to the researcher Stephanie Seneff, here I'll show you the next picture. What she's proposing, this lady, Stephanie Seneff, here's her book, by the way. Her book is called A Toxic Legacy by Stephanie Seneff, Toxic Legacy. And she claims that her research, she's been studying this for decades, suggests. She suggests that the glyphosate is substituting for a glycine, and this can occur in a protein's active site. It can occur in other locations on a protein. She claims that the glyphosate is able to get inside the cell, and it appears that this is what's happening. We're not sure for certain 100% if this is correctly what's happening, but this is what she suggests she thinks is happening. The glyphosate gets into the molecule like in the active site, and that in addition to having the hydrogen there, it'll have this phosphate projecting up into the active site, for example, or elsewhere the phosphate will be bulky, and it'll change the function of a protein, because a protein function of an active site it's a delicate thing. It's a question of, does it fit based on its size? Does it put the substrates in the correct proximity, in the correct orientation? Are they able to bind to it? If you've got this big, bulky, negative charged phosphate sticking into an active site that doesn't belong there, it's taking up space, it's producing charge that's likely going to repel things that aren't meant to be there. So it can significantly cause a lot of dysfunction of the enzyme or of a protein that's for other purposes. So that's kind of the main point of this talk, is to recognize what seems like a minor thing could be a very big deal potentially. So if you want to see Stephanie Seneff's book, Toxic Legacy, it's on pages 57 to 58 is the main part where she discussed this theory of hers. And I would say this reminds me of just the whole concept of a Trojan horse, whereby one molecule can mimic another and then get itself incorporated into other macromolecules, proteins, and other structures, and then cause them to dysfunction. F- can replace hydrogen. They're both very small. That's how I remember that one. Cadmium can replace calcium. I remember that because they both start with the letter Ca, cadmium and calcium. Aluminum can mimic some of the things that magnesium does. So magnesium is like this wonderful ion that we tend to be deficient in, and aluminum is like a disaster. So anyways, that was the concept. Trojan horse, inhibition of enzyme active site, proposed by Stephanie Seneff, and a general concept that can occur for multiple different toxins that can be useful to know about. Hope it's helpful.